Welcome to Bosker Sciences. Today's topic is going to be over energy and enzymes. There are 5 main topics that are going to be covered in this video. Number 1 is going to be the laws of energy. Number 2, metabolism. Number 3, energy-controlled reactions. Number 4, enzyme characteristics. And number 5, enzyme inhibition. If you've been watching my videos, you know that all living organisms require a constant input of energy. That's why biological systems constantly transform this energy from one form to another in order to carry out all life functions. There are multiple laws of dynamics, and the first one is the first law of thermodynamics, and that states that energy cannot be created or destroyed, only transformed from one form to another. This is also known as the law of conservation of energy. The second law of thermodynamics states that during energy conversions, the universe becomes more disordered. In other words, entropy increases. We can determine how much free energy is available to do work within a cell by calculating the Gibbs free energy, or just free energy, represented by the letter capital G. During the course of an interaction, if energy is released, the reaction is said to be exergonic or exothermic, and the change in G is negative. If energy is absorbed, the reaction is energonic, and the change in G is positive. In complex cellular processes, exergonic and energonic chemical reactions are coupled. That means exergonic reactions power the energonic ones. If you look at the graph on the right, you can see that it's an exergonic reaction and that the change of G is less than 0. So that means that energy is lost. That's why the products are lower than the reactants. Meanwhile, energonic reactions have an increase of energy, which is why the products are greater than the reactants. Since we got a basis for energy, let's learn about metabolism. Metabolism can be defined as the sum of all chemical reactions that take place in cells. There are 2 types of metabolism: catabolism, which is the breaking down of molecules, and anabolism, which is the building up of molecules. Metabolic reactions take place in a series, or pathway, each of which serves a specific function. These multi-step pathways are controlled by enzymes and enable cells to carry out their chemical activities with remarkable efficiency. If you don't understand the definitions between catabolism and anabolism, you can take a look at the picture on the right, and you could see that catabolism is breaking down larger molecules into small subunits, while anabolism is combining the small units into one larger unit. Remember that an enzyme does not provide energy for a reaction, and do not enable a reaction to occur that would not occur on its own. Enzymes serve as catalytic proteins that speed up reactions by lowering the activation energy, or the amount of energy needed to begin a reaction. In the potential energy diagram, the potential energy of the products is less than the potential energy of the reactants, so energy is released and the reaction is exergonic. The dotted line shows the same reaction when an enzyme is introduced. The enzyme serves to lower the activation energy, and the reaction proceeds. There are many characteristics of enzymes, but they can be summarized in a few bullet points. Firstly, enzymes are globular proteins that exhibit the tertiary structure. If you don't know the difference between protein structures, I suggest that you look at my 4 biomolecules video that I'll put a link in the description. Number 2, enzymes are substrate specific, which means that only a specific substrate can fit in the enzyme, like in the diagram. Number 3, the induced fit model describes how enzymes work. As the substrate enters the active site, it induces the enzyme to alter its shape slightly, so the substrate fits better. The old lock and key model was abandoned because it falsely implied that the lock and key were unchanging. So you should not think about the lock and key model, which you learned in normal biology. Number 4, the enzyme binds to a substrate, forming an enzyme-substrate complex. Number 5, enzymes are not destroyed during a reaction, they are reused. Number 5, enzymes are named after their substrate. and the name ends in the suffix ase. For example, sucrase is the name of the enzyme that hydrolyzes sucrose, and lactase is the name of the enzyme that hydrolyzes lactose. Enzymes catalyze reactions in both directions, and enzymes often require assistance from cofactors, which are inorganic, and coenzymes, which are usually vitamins. The efficiency of the enzyme is affected by the temperature and pH. Average human body temperature is 37 degrees Celsius, which is optimal for human enzymes. But when body temperature is too high, enzymes will begin to denature and lose their unique conformation and their ability to function. Gastric enzymes work better at a low pH, which is an acid, and then when it's mixed with stomach acid, while intestinal amylase works best in an alkaline environment, like the intestines. You can see the diagram that most human enzymes work better at a low temperature, while thermophilic bacteria work at a higher temperature, and the acidity is determined by how many hydrogen ions, which you can go back to one of my old videos, which is called 4 biomolecules again. In the second box, you could see that at a low pH, stomach acids work better because they are an acid themselves, while a high pH is a good environment for intestinal enzymes. Cell metabolism is tightly regulated by gene expression on enzymes, and this is done by inhibition. There are two types of inhibition. First is competitive inhibition, and that's when a compound resembling the original structure reduces the amount of product by preventing the amount of substrate that can bind to the active site. And this can be overcome by increasing substrate concentration. And the other kind of inhibition is non-competitive inhibition, and that's also known as allosteric. And that means that it depends on the shape, and regulators bind to an alternative site and causes a change in enzyme shape. You should also know about feedback inhibition, which is the end product of the pathway, is an allosteric inhibitor for an enzyme that catalyzes an early step in the pathway. In a normal reaction, the substrate would just go to the active site of the enzyme and get finished products. But in competitive inhibition, an inhibitor goes to the active site, which blocks off the substrate that's supposed to go there. In non-competitive inhibition, it goes to another active site, and that changes the shape of the enzyme, so the original substrate can't be turned into products there. That's the end of today's video, and I hope to see you guys next time.